Hi everyone. In this section, we are going to deal with agglutination reaction. So in previous section, we already discussed about precipitation reactions and its various types. Um, in this section, we are discussing another type of antigen antibody interaction, that is agglutination reaction. So this reaction is between the antibody and particulated antigen. In case of precipitation reaction, it is an interaction between the soluble antigen and antibody in an aqueous solution, whereas in agglutination reaction, this reaction takes place or interaction between the antibody that interact with the particulated antigen. That is, it is coated. This antigen is coated on some surface like lactose, etc. And in this reaction takes place at specific temperature and pH. That is very important. This takes place at a specific temperature and pH. As a result, visible clumping is formed, or we can say a visible clumping. So this reaction is named as agglutination reaction. And as a result, antibodies that produce or shows this reactions, they are called agglutinants. So the antibodies that produce these reactions, they are called agglutinants, and this reaction occurs at optimally. That means there will be or the it is when the antigen and antibodies react at a equal proportion. This agglutination reaction takes place, and agglutination reaction is similar to that of the principle of precipitation reaction. That is, when excess of antibody is present, it will inhibit the agglutination reaction. So this effect of mechanism or this mechanism of inhibition can be called as Proton effect. So Proton effect is that when excess of antibody is present, it inhibits the agglutination reaction. That is, this mechanism of incubation is called as Proton effect, and this effect can, or the Proton effect can be used in many types of immunoassays. This forms a basic phenomena, which Proton effect forms a basic phenomena in many immunoassays, and this effect is readily diagnosed by performing an assay at a variety of antibody or antigen concentration. Now, why this Proton effect is occurred, or how it is taking place. There are two reasons. First is when high concentration of antibody is present, or high antibody concentration, the number of antibody binding site is large in number comparing to the epitopes of the antigen. So, as a result, antibody binds with antigen only by univalent instead of multivalent. So that is because here the antibody concentration is very high. So they will only bind in univalent with the antigen. So as a result, it may fails to form the lactose agglutination. And another reason is that on using a polyclonal antibodies, the Proton effect can also occur in same way. So, because of this, the antiserum may contain, antiserum may contain high concentration of antibody that binds to the antigen that fails to induce the agglutination. Such antibodies are called incomplete antibodies. So, the antibodies that fails to induce the agglutination, they are called incomplete antibodies. And IgG antibody may act as an incomplete antibody. That is, when at high concentration, the incomplete antibody may occupy most of their all antigenic sites. So this will lead to blocking of IgM-SS. This IgM is a good agglutin, and at a high concentration of this incomplete antibody, it blocks the access to the IgM. So that's what the incomplete antibody IgG does here at a high concentration. Now, and it is not seen when agglutinin monoclonal antibodies, and this is not seen in agglutinin monoclonal antibodies. And this incomplete why this incomplete antibodies lacks their agglutinin activity? That is because due to two reasons. First one is due to the restricted flexibility in their hinge region. Their hinge region is restricted for the flexibility. That's the first reason. And the second one is maybe due to the density of the epitope distribution or the location of some epitopes in the deep pockets of a particular antigen. So the second reason is on the epitopes on the particulated antigen may be densely is based due to the density of distribution, or sometimes some epitopes may be present in the deep pockets where this antibodies can't reach for binding. So these are the two reasons why they lack the agglutinin activities. Incomplete antibodies lack this agglutinin activities, and this make it difficult for the antibody to assume the required angle for the optimal cross-linking of epitopes on one to two or more particulated antibodies. So due to these two reasons, this will makes the antibody or fails to make their cross-linking or optimal cross-linking of epitopes with two or more particulated antigens. And we can solve this problem by trying different antibodies that may react with other epitopes of the antigen that do not form these limitations. So by using this different antibodies, we can solve this uh, problem of incomplete antibody. And there are different and this agglutination there are different forms of agglutination which can be used in diagnostic tests, the which includes lactose agglutination, flocculation tests, direct bacterial agglutination, hemagglutination. These are some of the different forms of agglutination reactions which can be used as a diagnostic test.